So Simon Griffith who's joining us today, has been at Macquarie University since 2007, where he leads a research group focused on the evolutionary ecology of birds. And today he will be telling us specifically about speciation in finches and the conservation implications of that. His research niche has been understanding the determinants of evolutionary fitness amongst individuals within populations in the context of mating, parental and social behaviour. And an important focus of work has been the well-worked zebra finch. I like that term, well-worked, with Simon establishing and running the longest population study of wild birds to help contextualise the lab research focused on this species around the world. So thank you so much for joining us today, Simon. I know really nothing about finches or birds in general, and I'm quite excited to hear your talk. So thanks for joining us, and the floor is yours.
Okay, thanks very much. Can you see my screen and hear me?
Yes.
Yep. Okay, excellent. All right, thanks very much for the invitation to talk to you guys today. I will start out by saying that I don't really work in the area of conservation. So since I gave you my title, I've now put brackets and a question mark around the conservation part. And I'm interested in getting some feedback from you guys at the end of my talk as to where the contributions may come in that area. So I've done a lot of work in the zebra finch. So for 16 years, we've been running a long-term project on zebra finch, both in the wild, so a field site based in far west New South Wales, and also in the lab. And the zebra finch is one of the most studied birds in laboratories, and most of that work is happening in the northern hemisphere in Europe and North America. The reason for that is because they're very easy to work on in the lab. So they're kind of the lab rat equivalent for birds. So we can use zebra finches to understand lots of questions in genomics, neurobiology, behavior, etc. So they've been very well worked, as I said, as was said in the introduction. So current work on the zebra finch is actually focused around the resilience to heat. So we're using them as a model system to understand how birds adapt to an increasingly extreme climate. One of the research areas where I've done a lot of work, but the zebra finches aren't particularly useful, unfortunately, is asking questions about speciation. And the reason for that is that the zebra finch has a panmictic population across Australia. So there's essentially no genetic structuring at all in that very big population of birds. And there's no subspecies or even races within the Australian continent. So as a result, for these two questions that I've always had a long fascination with, what is a question and what are the mechanics of speciation? We followed so the zebra finch is obviously a very useful species to work in labs, and that's because of many of the characteristics and the ease with which you can hold finches in captivity. So we chose to work with a couple of other finches which are closely related and almost as easy to look after in the lab, but which have much more interesting genetic structure, and we can work on both in the field and the laboratory, where we can conduct more manipulative work. So we all know what species are and how we can recognize them. So whatever flavor of animal or plant you are studying or interested in, you can go and buy a guidebook from a shop, or these days you can download it from an app store. And you can relatively easily identify what we call a species. But of course, for many species, there is a lot of variation out there that we know about, and a lot of that variation is captured in the books. So for example, here on the right, the very aptly named varied Citella comes in a variety of different forms across the continent of Australia. And I'm interested in understanding why we have all those various forms, how they interact with one another, and what processes maintain those forms since they've come back into contact after whatever processes gave rise to them in the first place. Now, the varied Sotella would be an absolute nightmare of a species to work on because they're actually very difficult to see and find. And for that reason, again, following the example of the work on zebra finch, we chose to focus on a couple of relatively well-known and birds that are held in captivity, that are very commonly held in captivity. And also, importantly, these two species have very strong genetic structuring, but in two different ways. So firstly, we have the Gouldian Finch. So this is a species with very distinct morphs that are genetically distinct. So we have black-headed morph, a red-headed morph, and yellowheads. And in these pictures here, these are all males, but the females are essentially a kind of duller version of the same color scheme. And then a similar, and actually coincidentally, we also have in this system, we have yellow, red, and black, which may be a coincidence or perhaps maybe not. But here, instead of being genetically discrete morphs, we have variation across a geographical cline. So these are the long-tailed finch on the left, yellow-billed and red-billed long-tailed finches, and then the black-throated finch on the right-hand side, or distributed mostly in Queensland, or probably almost entirely in Queensland. So these two species offer us the opportunity to look at how species form and the mechanisms that, that gave rise to them, and also enable them to persist in these different forms. So the first work we did was on the domesticated Gouldian finches. So this was work that was done in collaboration with a postdoc, Sarah Pryke, and we worked on domesticated birds, and we had a number of outcomes from that work. And I've just summarized a couple of those here. So importantly, we found that these genetically discrete morphs are incompatible with one another to a reasonably significant extent. So a black-headed female that you can see on the bottom here, if a black-headed female pairs with a red-headed male, so the head color mismatches, the sons will be 40% more likely to die, and the daughters 83% more likely to die than those of pure partners or pure pairs within the first couple of months of life. So we have a very significant incompatibility. There's lots of life history adjustments. So females in mixed pairs reduce their investment in these kind of mixed poor quality matings. And the genetically discrete head morphs also are co-vary with other traits that are discrete amongst these the different morphs. So the red morph is much more dominant and aggressive than the black-headed morph, and the red morph dominates the black in social interactions. And these are birds that are very social. They tend to aggregate in often quite large flocks and large groups. So they move around, they breed closely together, and they forage closely. So pulling together all of the work that Sarah did over a number of papers, we pulled all the bits and pieces of that explained the life history and the dominance interactions and the genetics. And Hannah Coco built a model where she demonstrated some complex hawk-dove dynamics in this system and a density dependence that contributes to a stable equilibrium, such that we have the persistence of these genetically discrete morphs in populations. And so if we go into the wild, both historically, so on the left here, we have the historic frequency of black-headed and red-headed birds, both in males and females. And we tend to find that the frequency is relatively stable spatially and temporally. So males, about 60% of all males are black-headed, and about 40% of the remainder are red-headed individuals. In females, the frequency of blackheads is much higher, around about 80% on average across populations. And we find the same frequencies in populations today. One of the consequences of these discrete differences between morphs is that in very small populations, the model that Hannah made shows that in small populations, the equilibrium is disrupted, and often one of those morphs will drive the other to extinction. So from a management or a conservation management perspective, these morphs interact agonistically and predictably with each other, and they can actually be thought about as a threatening process to one another. And of course, if you lose one of those morphs from a population or potentially the species as a whole, then to some extent the species isn't intact with respect to all of the standing genetic variation. So the morph frequencies certainly need to be accounted for in small isolated populations and certainly in reintroductions. So since Sarah left and we stopped working on these birds in captivity, but we've continued to do work on the genomics of these different morphs and also some conservation or population genetics. So the main goals of that, so this was work that was done with a PhD student, Perry Bolton, in my lab. The main goals were to explore the scale of the bottleneck as the species declined, understand the dispersal dynamics through the genetic structuring, and examine the molecular evidence for the incompatibility and the genetic divergence between the two morphs. So we did this by working with just over 100 skins that were collected from 1890 to 1920. So these were effectively represented the populations probably before most of the kind of landscape disruption happened across the top end. And certainly before the reported decline of the species, which began in the mid-20th century. And we compared that with 250 contemporary individuals or samples from those. And that was done in collaboration with Sarah Legg and Kim Mort at the Australian Wildlife Conservancy. So we used a number of different molecular markers and loci, microsats, mitochondrial DNA and SNPs to compare these. And again, I'm just going to rush through the results that we found. So the most important result here was that although we have these genetically discrete morphs, overall, across both the historic and contemporary populations of Gouldian Finch, despite the fact they're spread right across the top end of Australia, we found no evidence of any genetic structuring in either historic or contemporary populations. That does suggest that they're a highly dispersive species and that birds are moving around probably on an individual level each year, perhaps as they search for abundant sources of food. We also found no evidence of a significant bottleneck or decline in genetic diversity across the 20th century. So this was a bit of a flagship for endangered birds in Australia for a number of years, and it was reported that the decline in the 20th century might have been as high as 90%. If it was that high, we would definitely have found evidence of a significant bottleneck, but we found none. So I think we still need to better understand the decline, but it's possible that this species, which is still relatively, it's not very abundant anywhere in the landscape at the moment, it's quite possible that it was always a species that was relatively rare and little encountered. Interestingly, though, we did find that there was no structure between the red and the black-headed morphs in the wild, apart from at a very small region of the genome. But there is structure in the domesticated populations. And there's also clear structure between the domesticated and wild population overall. And interestingly, the genetic incompatibility that was so significant in the domestic population that we studied, there's no evidence of that occurring in the wild at all. So what this kind of suggests to us is that as these birds have been brought into domestication and managed by aviculturalists for many tens of generations, that a combination of different processes has actually changed this species from the wild birds that were brought in initially. So we know that aviculturalists modify populations, and we have a paper in Press in Emu at the moment that has measured this genetic change, but also surveyed aviculturalists about their practices. So they exert artificial selection on traits such as the color of females. So all domesticated females are much more brightly colored than their counterparts in the wild. Furthermore, aviculturalists typically will only keep either red or black birds, or if they keep both, they'll keep them and breed them quite separately in different cages or aviaries. So that means that a number of different population processes have probably led to the divergence of the different morphs in the domesticated populations, and possibly have given rise through some process that we as yet don't understand, which has actually driven that genetic diversity, the difference in the genetic diversity. So again, there's a number of conservation challenges that remain to be addressed in the Gouldian finch. How do we measure the decline of a highly mobile species? So a lot of the work, survey work at the moment on Gouldians is based on waterhole counts at a few specific places across the landscape. But when a species is highly mobile, that can be a problem. I'm still not sure that we understand the historic abundance of the species, and that, of course, is what we're judging the decline and the status of the species against. And then there are also questions about reintroductions. So this bird is very abundant in captivity, and there have been introductions of captive bred birds or domesticated birds back into the wild in a number of attempts over the past few decades. But again, I would suggest that we are very careful in the provenance of birds that are introduced into the wild, given the demonstrated divergence between captive and wild populations. So long-tailed finch offered another alternative to understand genetic divergence and the speciation process. So again, here we have two subspecies of the long-tailed finch. In the west, basically centered around the Kimberley, we have the yellow-billed form. In the center, we have the red-billed form, and then in Queensland, we have the black-throated finch. The black-throated finch diverged from the other two about 0.6 million years ago, and the other two diverged about 0.3 million years ago. So the obvious place to start this work was to go and collect samples or go and measure birds in the wild and measure bill colour, because bill colour was the morphological trait by which the species were characterised. So we measured birds across 26 populations, covering about 80% of the whole range of the species from west to east. The plot in the middle is a plot of the individuals and the bill colour plotted in colour space. So what we find is in the east part of the range, all the birds have pretty much the same expression of red. In the western part, the birds have a fairly uniform variation or lack of variation in bill colour, and they're all yellow. But we have this zone of around about 150 kilometres in the middle, and it's actually right in the middle of the Northern Territory, where we find these birds with these intermediate beaks. So again, the Gouldian finches are either redheads or blackheads. Here we have a continuous trait, and that suggests that there's a number of genes involved. And with collaborator Daniel Hooper, based in the US, we've been looking at the genomics of these traits. And we've identified 3 candidate genes based on genome-wide association mapping for bill colour. One of those is on the Z chromosome, which is the male sex chromosome in birds, and the other 2 are on chromosome number 8. And these 2 genes here have both been identified as responsible for colour variation in both the zebrafinch and also in the plumage of the canary. The nice thing again about finches is that we can bring them into captivity. They're easy to look after, they're easy to breed. And so we can actually, we've generated a number of hybrids and back crosses to enable us to further explore the genetics around colour and other traits. So here are the results of some our lab-bred F1 hybrids, and these are either female or male offspring produced by these pairings. And the pairings to produce F1 hybrids can either have a red female with a yellow male or a yellow female with a red male. And what we find is that all of these F1 hybrids have intermediate colour, so they're all orange. but where they've inherited a Z from the yellow father, those females are more yellow than the females that have inherited their Z from the red father. And that makes sense because we know that one of the genes is on the Z chromosome. So we're also doing work with what we call long black crosses. So we're crossing black-throated finches with both the red and the yellow long-tailed finch. This is a picture of an adult, one of those. And as you can see, they also have an intermediate bill with a black tip and an either kind of reddy orange or a yellowy orange base to it. And so this work is continuing. Maybe I'll report on that at some point in the future. So bill colour is obviously a trait that an individual in a population where two different forms are in contact with one another, an individual can identify through the process of mate choice, can identify a member of the opposite sex which belongs to the right species as opposed to a member of the opposite sex from an alternative species. So this helps us to understand the barriers to species basically breaking down. So this would reinforce the difference we're finding between the yellow birds and the red birds, even though those have probably been back in contact for several 100,000 years. Another isolating mechanism that we can think about are the post-copulatory ones. So here, these are pictures, scanning electron micrographs of sperm from the different species. And so we've also been exploring the potential for post-isolating mechanisms. So if a female can't actually identify very effectively the type of male that she's breeding with, then perhaps the lack of fertility of that male partner will prevent the spread of genes from one form into the other. So we've found that there are significant differences in the morphology of sperm between both longtails subspecies and also between the longtail and the black-throated finch. And in mixed pairings, which again is something we can do in the laboratory, we find that, so this is the total on the y-axis here, we have the total number of sperm which are captured on the the yolk membrane around the egg at the time of fertilization. So when one sperm penetrates and fuses with the ova, the acrosome reaction effectively locks all of the other sperm that almost fertilize that egg into the yolk membrane. So through this way, we can actually count the number of sperm that have traversed all the way up the female reproductive tract and almost fertilized the egg. And what we find here is that the sperm of a pure pair mating or a male in a pure pair cross are far more effective at getting all the way to the site of fertilization than in these two different back crosses. And previous work on the zebra finches, we've also found that the genes underlying variation in sperm morphology are largely, most of those genes are found on the Z chromosome, which again is the male sex chromosome in birds. So here we have some Manhattan plots comparing the levels of divergence across the whole genome, across the different chromosomes, when we compare firstly populations within each of the subspecies. So at the top here, we're comparing the full extent of variation captured in the yellow populations and in the red populations. So here we have the variation across the genome over 750 kilometers of the range of the red-billed longtail. At the bottom here, we're capturing the variation just in within the hybrid zone. So the two populations on the outer edges of the hybrid zone between the yellow-bill and the red-billed birds. And the most striking thing you can see here is that most of the variation is on the Z chromosome. So in fact, 97.9% of all of the variation across the genome between these two forms is found on the Z chromosome. The reason that's the case is because the Z chromosome has two huge inversions in this species, and those inversions have locked together genes at some point in the speciation process. They've locked together a lot of genes in either the eastern form or the western form and those genes can no longer recombine. So essentially the Z chromosome has a very, very low rate of recombination. And so, so we find that most of the Z chromosome is inherited as a very large block, or a supergene, if you like. And you can see evidence of this if we look at the, so this is work that Daniel's, where Daniel's looked at the mean migration rate of different genes, both in the autosomes and also in the Z chromosome. And over in the east, we can see that the contact zone between the black throat and the red-billed long-tailed finch is very similar for both the autosomes and the Z chromosome. This brown area here is where there's a very low level of migration of genes in that part of the genome. However, if you compare the movement of genes within the autosome to the movement of genes within the Z chromosome, over on the eastern side of the Kimberley, we can see there's a significant reduction in the movement of genes on the Z chromosome across that area. So that suggests that there's something interesting happening in that particular area. So we've basically gone in and studied this contact zone and the area shown on this map. So we've gone and done some high density transect work, capturing and sampling, just blood sampling individuals across this, across a number of populations within an area of around about 200 kilometers. The important thing to note here that this whole area is to the west of the colour bill cline. So the colour bill cline is off to the east of here, and all of the birds in this area here all have a yellow bill form. So that means that any variation within those birds is probably cryptic. It's certainly cryptic to us in the field, and it's probably cryptic to the birds that are living in that area. And then we've used the genomic data that we've got to actually calculate the cline. And what we find is that the Z chromosome cline in that area is steeper than the cline for the bill color. So that means that as we change from the eastern to the western form, the bill color cline spans about 150 kilometers. The Z chromosome cline, as we move from the western form to the eastern form of the Z chromosome, that cline's only around 115 kilometers. So it's much narrower. It suggests that there's quite strong selection there. And this, as I say, importantly, these two clines are about 350 kilometers apart. So another important aspect of genomic architecture or genomic variation is variation in the mitochondrial genome. And once again, we find that there is a very clear divergence between eastern and western subspecies. So there's 22 mutations that separate the eastern and the western forms. But again, importantly, the eastern forms have come, they've moved much further to the west than the bill color cline. So again, in this area centered around Kununurra and Wyndham, where the Ord River comes in, we find lots of eastern forms or eastern mitochondrial haplotypes in that area. And again, there's a very narrow cline, which suggests there's very strong selection. And this is again some work done with Daniel Hooper and a student, Kelsey Lopez. So this is work that I'm not involved in at all, but I really encourage you to go away and read this work. It's some excellent work that's been done by Paul Sunnocks and his group down at Monash University. And this work is focused on mitonuclear ecology and evolution in the eastern yellow robin. And what they've found in a number of really amazing papers is that they've found very strong mitonuclear lineages that differ by 6.8%. By contrast, in that long-tailed finch data, the divergence we have is around 1%. But here, these two mito-lineages differ by 6.8%, and the contact zone between them is extremely narrow. So it's around about 20 kilometers. Again, importantly, eastern yellow robins that are holding either one of these two mito lineages are completely inseparable from each other, which means the birds themselves can't tell whether a potential partner has the same or a different mito lineage. The interesting thing as well about this mitochondrial divergence that Sunnox and his team have identified is that it's of huge functional significance. So although the mitochondrial genome is very small, a number of genes, there are about 9 genes on the mitochondrial genome, which are responsible for coding key elements in the oxidative phosphorylation pathway. And each one of those genes, so there's around 9 genes in the mitochondrial genome, each one of those genes interacts with about 6 or 7 nuclear genes. So they interact in concert with one another to move chemicals through the membranes of mitochondria. And of course, this is hugely important because the oxidative phosphorylation process is the effective engine room for driving the production of energy in cells. So any inefficiency that might be introduced by an incompatible mitochondrial genome and nuclear genome is likely to significantly affect the energy production of organisms, and that will have knock-on effects for lots of different functional traits. And of course, one of those is thermoregulation. So it's no surprise that they've found very strong links between this very strong cline and climatic variation in the area in which they've studied this most intensely down in Victoria. So here they've looked at temperature and precipitation and found very strong clines with respect to this the mitochondrial divergence. So in summary, our findings in the long-tailed finch so far suggest that we've got the kind of classic divergence between these two forms that was anticipated by looking at either museum skins or birds in the field. That is, we've got divergence on forms on the basis of bill color with a relatively narrow contact zone. But what our recent work has found in the last decade is that there's actually a much stronger contact zone, or 2 very steep clines, with respect to both mitochondrial divergence and Z chromosome divergence. That's completely separate from that bill color cline and located to the adjacent to the edge of the Kimberley Plateau. So we're currently further investigating the functional significance of that mitochondrial divergence and the Z chromosome divergence. And we're also further exploring its role in important functional traits for both pre and post-copulatory selection. So we're looking at sperm and song. So recently in the lab, we've been conducting breeding experiments with the red-billed form and the black-throated finch, which have a high level of divergence. So we should be able to see even greater effects on fitness. And if Anastasia ever lets me into Queensland, we're hoping to get a team up into the contact zone up in the Gulf of Carpentaria. So the work that we've done, I think I'm excited to place it in the context of the really cool work that was done on speciation by Hugh Ford back in the 80s. So here he mapped out, in this really classic paper, he mapped out the contact zones between the avian hybrids, so gaps between species and subspecies and races that were identifiable on the basis of really obvious traits like plumage. So these are the kind of things that you could identify from museum collections or even just looking in the bird book in many cases. And of course, these hybrid zones are reasonably well established and supported in relation to refugia during the Pleistocene aridification of Australia. What's really exciting is that in the past 10 years, the work by Sunnox and his team and our work has shown that if you actually go beyond these obvious morphological traits, there are really extreme points of divergence at different elements of the genomic architecture, which suggests that actually in many species, we're likely to see a greater number of hybrid zones as populations and populations vary spatially, and particularly with the extent to in relation to local adaptation. And of course, we have lots of declines in climate, both across the dividing range and also as we move up and down the East Coast. So the main conclusions are that species vary across their range and also between captive and wild populations. And this highlights the importance of local adaptation and genetic processes. And I think we're only just starting to uncover the extent of the variation, particularly in the mitochondrial genome and also in the structural elements of the whole genome for many species. And I think there are some implications here for species management. So certainly we're identifying the genetic incompatibility that might occur in line with the kind of genetic variation that we can see, and the morphological variation that I guess we've known about for a little bit longer. And the way that can cause functional breakdown between admixed populations, both in terms of fertility, but also in terms of really important physiological traits, such as the functioning of efficient mitochondria. So that means mixing populations needs to be done probably with great care. So we also need to understand the nature of local adaptation. And again, I think there's some really exciting opportunities to look at how fine local adaptation is in some of these cryptic elements here, and particularly with respect to a changing and more extreme climate. And I'd like to finish up by acknowledging funding from the ARC through a number of discovery projects that have supported this work over the years. Sarah Legg and Kim Mort through the Australian Wildlife Conservancy for their contribution of very valuable Gouldian Finch blood samples for the genetic work. And then current collaborators on this project are Daniel Hooper and Melissa Rowe. Daniel based at Columbia University in New York, and Melissa at the Netherlands Institute of Ecology. And finally, the 4 students who have made contributions through their PhD work on these species: Perry Bolton, Laurie Hurley, Kelsey Lopez, and Callum McDiarmid. And I'm very happy to take questions there. Thank you.